Good morning, everyone. My name is Callender, and I'm working with EUREKA 10, Professor Herbert Waite's lab in the molecular biology department. I'm a EUREKA intern here at UCSB, and I'm studying the effect of iron deposition on muscle thread strength. So the organism producing structures that I'm studying is Mytilus californianus, commonly known as the California mussel. It's a local organism that lives right off the coast here in Santa Barbara, and it produces structures known as byssal threads that it uses to attach to rocks in the intertidal zone. You can see them kind of right there. Byssal threads have evolved to be extremely strong, very extensible, and they also show a really amazing amount of recovery after being stretched. They can be stretched up to 100% or twice their original size without breaking, and then they show a good amount of recovery afterwards as well. So biologists are interested in how these threads are made and deposited. However, engineers are also interested in how these threads are made and their molecular structure, because knowing more about how they're made and produced could lead to the ability to design better synthetic materials. So pictured on the left are two byssal threads, and in these threads there's a protein side chain called DOPA. Pictured on the right are 3 DOPA side chains forming a crosslink with a metal ion in the center, specifically an iron ion in the Fe3+ form. And this is the specific type of protein-metal interaction that I'm studying the effects of in my project. So it's been proposed in past research that increasing the amount of protein-metal interactions in the threads can increase thread strength and toughness. So pictured on the left is an image of a byssal thread. They're composed of an inner fibrous core and an outer covering or cuticle, both of which have pretty different biological properties. Pictured on the right is a cross-sectional image of a thread, and you can see that while the iron is sequestered in the cuticle or outer covering of the thread, there's some, to a lesser extent, present in the core as well. And this kind of brings up the question of how much of a role do potential DOPA-iron interactions in the core play versus DOPA-iron interactions in the cuticle. In the past, researchers have focused on the thread as if it was one uniform structure, or they've only really looked at the cuticle as the primary location of these bonds that are happening. And that may not be the best way to be looking at this question. So that's what my project was trying to look at a little bit this summer, is the role of DOPA-iron bonding in the cores versus DOPA-iron bonding in the cuticles as you increase the amount of DOPA-iron bonding that's taking place. So the way I did that was by soaking threads in solutions of different iron trichloride concentration, mechanically testing those threads to establish a correlation between thread mechanical properties and increasing iron DOPA bonding. And then I hope to eventually use chemical visualization methods to see where the iron's actually going in the thread and to what extent it's being localized in the cuticle or the core as you increase the amount of iron in the thread surroundings. So this is what I did, my setup for the summer. I harvested threads from live mussels, and then I soaked them in solutions of varying iron trichloride concentration, specifically 1, 10, and 100 micromolar iron trichloride. I made these solutions using a buffer that was set to be at a pH between 5 and 5.2, and the reason I did that is because I wanted the iron to be in a proper form so that it could be absorbed by these threads and form the crosslinks with the DOPA. So I was wanting it to be in the Fe3+ form, which happens at that particular pH. I soaked the threads for around 12 to 24 hours, and then I mechanically tested them using a tensile testing machine, which, pictured right here, it's basically just 2 clamps that grab either side of the thread sample and stretches it at pre-programmed speeds and with various parameters that I enter in. All the while, this machine records various mechanical values for the thread that I can use to characterize how the iron treatments have affected its mechanical properties. So just 2 of the main values that I get out of this machine are stress and strain. They're pretty simple mechanical values. Stress is the force experienced per unit area, cross-sectional area, as the sample is stretched. And strain is the change in sample length divided by the initial sample length. So this is an example of a graph that I get out of the mechanical testing process. It's a stress versus strain graph, and essentially what I told it to do is to stretch the thread to around 40% strain, and to then relax and let the thread recover for about 60 minutes. And then it repeats the cycle a second time. So one of the important pieces of information I can get out of this graph is the peak stress of the material, which is where the thread has started to deform in response to being put under the strain. Another important piece of information I can get out of it is the level of deformation, which is essentially the gap between the first and second cycle. So that's the percent strain that the sample was unable to recover in the 60 minutes that it was just sitting there in the system. Another important thing that I can get from these graphs is the modulus of the material. The modulus is stress divided by strain. So for this graph, it's simply the slope of the initial linear portion of the graph. And it's considered to be a measure of stiffness of the material. So in terms of my results, I got some results that I was expecting and some results that I was not expecting. So for the modulus of the first cycle and the second cycle, you can see there's a lot of variation present. And the averages between the different iron treatments are not significantly different from one another, which is not what I was expecting to happen. Similarly to the first cycle, the second cycle modulus are all pretty much the same for the different treatments, although you can see there has been a significant decrease due to damage to the thread after being stretched in the first cycle. Similarly to modulus, the peak stress of the material, there's a lot of variation, and not the variation I was looking for, which is variation between different iron treatments. Level of deformation, however, did show significant variation between different iron treatments. Specifically, the two middle treatments, 1 micromolar and 10 micromolar, showed a significant drop in data average when compared to the control and the 100 micromolar treatment. So in general, the data averages for the first and second cycle for modulus were not significantly different between different iron treatments. Same with peak stress, data averages were not significantly different. For level of deformation, which was the gap between the first and second cycle, the data averages for 1 and 10 micromolar treatments were significantly different from those for the other ones. So due to all the variation in my data, I wanted to go back and kind of make sure that the chemistry that I was assuming was happening actually was happening in my solutions. Namely that the controlling of the pH was enough to ensure that the iron was bonding with DOPA in the threads. So I took a sample of threads and I put them through the exact same protocol that all the other samples had been through, except I didn't stretch them. Instead, I used energy dispersive X-ray spectroscopy to chemically analyze what elements were present in the thread. And so I was specifically looking for iron. And as you can see, the buffer, which is the control, showed 0.183 and 0.455 weight percent of iron. And then all the other treatments showed, while it's still increasing, not too different from the control. And with EDX as well, any weight percent below 1 weight percent, it starts to become questionable as to the accuracy of the reading. It's just such a small amount of iron. So essentially, the iron may not be bonding with the DOPA in my treatments, or if it is, it's at such a small level that it's deemed insignificant by the level of accuracy that EDX can give you. So what I hope to do next is to repeat this EDX analysis just to be sure that what's happening is that the iron is not bonding with the DOPA in the threads. And if that's the case, I need to reevaluate the setup of my experiment. If there's oxygen present that could be causing the iron to be oxidized, that could be putting it into a form that makes it so it can't form the crosslinks with the DOPA. And I can try and think of methods to remove as much oxygen as possible from my solutions. Eventually, I hope to move on to the visualization of the iron localization to see where it's going in the threads, and potentially more detailed mechanical tests such as nanoindentation may provide me with a more controllable mechanical test of certain spots on the threads. I want to thank EUREKA, Herbert Waite, everyone else in the Waite research group for helping me out with my project and providing so much support this summer. And I also want to thank Erica Lubin and everyone else with the EUREKA program for helping me out so much as well, the Center for Science and Engineering Partnerships and everyone involved with that group. Thank you.
When you did the testing, did you actually break the strands at the end?
Well, when I was taking them out of the machine, I did, yeah.
Okay.
But not for the actual measurements, because if it was actually breaking, the strand would look like it had stretched, and then it would keep going, and then it would just drop off from when it broke. Yeah, so I wanted to stretch it to the point where I could see the peak stress point, but I didn't want to break them, because I wanted to observe how they could recover in between cycles. Yes?
You said you were working in pH 5-ish, right?
Yeah, about between 5 and 5.2.
Is it safe to assume that the mussels themselves wouldn't be at such a pH? And do you know how you could account for such a difference in the pH?
I'm not sure how mussels would react in that pH, but I was not actually soaking the mussels themselves in my iron treatments. I would harvest the threads from the mussels and then soak them in the solutions. I don't think they would like being in a pH that low, but they're pretty hardy organisms, so I don't know how they would react. But they're usually used to being in a pH of around 7 or 8, which is the pH of seawater. The reason I lowered the pH is because at 7 or 8, the iron would not be in the Fe3+ form. So I wouldn't be able, I wouldn't be able to assume it was being absorbed by the threads. Yes.
I have a couple of questions. So,
Mhm.
you said that possibly oxygen could be in there to oxidize the iron. But, I mean, if they were in seawater, there would be oxygen present.
Yeah.
So, in the natural environment, when they're attached to the mussel, does the iron actually come directly from the water, or is the thread processed inside of the mussel and then grown almost like hair?
That's a really good question, because it doesn't come directly from the water. So
Okay.
in the wild, what happens, because it's at the slightly basic pH of seawater, and the iron wouldn't normally be in the form where it can just directly bond with the dopa, the mussel has found a way to work around that as an organism.
Okay.
They filter feed, so they take in the ocean water as part of their feeding process, and they, I'm pretty sure we think that they take the iron in and they convert it into a usable form, and then they do, I don't know if it's really like hair, but they do deposit the threads and make them. So that could be how the organism has worked around it in the wild.
So would it be more reasonable, could you run a very similar experiment but expose the mussels to higher concentrations of iron and allow them to naturally
Yeah.
process the threads and then test them? Because then you wouldn't be dealing with having to change pH, you wouldn't have to worry about the oxygen levels. It seems like it would almost eliminate some variables
Yeah.
as long as you had a large sample size of different mussels to make sure that they're
Yeah, I
wasn't all
originally
variants.
didn't want to involve the organism because I thought that would introduce variables.
But it might be required to
Yeah,
get iron
and I
from
thought,
the thread.
maybe I can reduce the variables just by looking at the thread as a material and just harvesting it and just eliminating that whole organismal aspect. But if that's the only way that iron can go into the thread, then that may be the only option.
It might be even interesting to investigate how they actually do that and then possibly find a way to do that sometime.
Yeah. Yes.
So did you guys consider using other iron resources instead
Yeah.
of iron trichloride, maybe more,
Yeah.
maybe more soluble or maybe more different forms of iron source that you could maybe it absorbs a little bit easier?
Yeah, there were other sources of iron 3 that I've very recently been considering. I've yet to go and see, what we have available, what I would have to order if I wanted to use different sources of iron. But there may be some better options besides iron trichloride, definitely.
And I guess the follow-up question is, in your elemental kind of analysis for how many irons are there, is there a way for you to determine what the maximum amount of iron? So if your thread, is there a way to know how many DOPA coordination sites
Yeah.
are actually present so that you would know the maximum amount of iron that could be there?
That was another thing I was thinking about, that there should be a maximum amount of iron that can be absorbed by these threads. And so I guess, you could just keep bombarding them with iron and see how much they would absorb. The solutions that I'm using are pretty low concentration. They're already much higher than the normal concentration in seawater, which is a very, very low concentration of iron. So that would be another interesting thing to see is what the limit is and if it eventually has any negative effects on the mechanical properties of the thread.
Maybe one last.
Yeah.
Sorry, it's really interesting.
Ask as many questions as you want.
Now, if you do want to eliminate the organism, is there a way that you could chemically expose the threads to some sort of condition that would allow them to, the bonds with the iron to break, and then so that they're try and deplete them of their natural iron concentrations and then expose them to concentrations and see if then that would be
Well, there has been research done in the past where they reduced the amount of iron present in the threads
Yeah.
by metal chelation. So they reduced the amount of iron, and then they did mechanical testing, similarly to what I'm doing, only reducing the amount of iron present.
And
So
then
that is possible. And then
Yeah.
you mean to like reduce the amount of iron and then to add the iron in?
Yeah.
Yeah, that might provide more control over the amount of iron.
Because if they're already at their maximum, no matter how much you expose them to,
Yeah.
they're probably just won't absorb.
I thought about that. I'm not sure if they're at their maximum at this point, because there is someone else in the lab who was using the organism to study them. And I know that he was seeing with the organismal threads, iron was going in, and it was at a higher concentration than the solutions I'm using. So I considered that, and I'm still looking into that, but I'm starting to think that it's probably not because it's saturated, like with dopa iron interactions. Great. Thank you.